Welcome everyone to our November Lunch and Learn. Today we're going to talk a little bit about microbial control strategies. So a lot of what I'm going to talk about, I've probably talked a little bit about on other Lunch and Learns before. And today's presentation is more about reframing things and getting you to think in maybe a little bit of a different way. There's probably lots of strategies that you're already doing, you're already using. There's a couple of interesting ones that I might tell you about towards the end of the presentation in terms of using microbial species as control species and control strategies. And some of that, again, is already happening in your fermentations or wineries. So it's just a matter of trying to us to think in a different way and realize that grapes and wine are very complex mixtures, and there's lots of things going on, and everything, when we are talking about yeast eating up sugar to convert it to alcohol, that's a very straightforward process. We think of it as a very linear process. There's a lot of other things happening while yeast is eating up that sugar. And there's a lot of interactions that can happen in the wine juice or wine matrix or even grape on grape surfaces and things like that. So just trying to make people think about those things and be thoughtful about those things. So let's jump into it. There's different ways that we can control what microorganisms we have on our grapes, in our juice, in our wine. And the three types of controls I will talk about today that will lead us to quality wine. And what does that mean? That's kind of a subjective term, or I guess it's an objective term that you can, you define at your own winery what quality wines are. But usually we have some sort of chemical control that we are performing. There's physical controls that we use, and then this idea of microbial control may be a little bit new. You're definitely doing some of it anyways with your winemaking, but we'll get into what do I mean by microbial control in a minute. So we need, the one thing I want to stress is we talk a lot about sanitation and cleaning and preventing spoilage, and we want to talk about getting rid of microorganisms a lot. We need some of these microorganisms in order to even have the products that we sell. So they're really important, and it's really important to understand that there are relationships between microorganisms we want and microorganisms that we don't want in our grapes, juice, and wine. So some of these microorganisms, so on the left-hand side here, just some cultured species. These are what Saccharomyces, so the first little square, that's what it might look like if you grow a lot of it on a plate, and that's what all of these are. If you grew a lot of these microorganisms, this is what we would see with our eyes. Obviously, when they are under a microscope, a lot of them look different. A lot of them are just small ovoid type single cell organisms. But there's a large variety, I would say, of yeasts that we can have in our wines, and they come in a lot of times on the grapes, or they live in the wineries, let's say. Thankfully, wine in general does not allow the growth of pathogens, so wine is typically safe. And so these microorganisms like these yeasts that are growing, and potentially some bacteria, most of the bacteria that we see are lactic acid bacteria. The microorganisms that we see growing or surviving, let's say, in juice and wine, are more so they're not pathogens. They're not going to hurt us. They may do some damage to the wines and make the wines not very pleasant, but they're not going to hurt us. So just some examples of ones that we might not want to see in our wines. The Saccharomyces, obviously we use that a lot, but there are lots of different species of Saccharomyces. On the right-hand side is a picture. I showed this picture at a presentation I did earlier this year. And so what is going on in this picture is there are 4 separate fermentations, okay, A, B, C, D are the different fermentations at the same winery. And so they investigated the fermentation at different time points. So CS is the cold soak, so that's before the alcoholic fermentation. Then the early alcoholic fermentation is ER, M is mid-alcoholic fermentation, and F is the end stage of alcoholic fermentation. And so they inoculated A, B, and C with a common yeast strain. They inoculated with this RC212. Okay, so they added that in as their culture, as their inoculum, like many of us do. And then they followed what species were present in the fermentation as the fermentation went on. The last letter D, the tank for letter D, they didn't inoculate at all. They just let it go wild. So the thing to point out here is that you have, besides letter C, the tank with letter C, you really don't have that RC212, that yeast that they inoculated with, taking over as the main microorganism in the fermentation. There's lots of other things going on that could be due to the tank was fermented in. It could be due to the grapes that the yeast that were present on the grapes. I will say you could see that the blue bar at the beginning of the fermentation before they added the inoculum is Hanseníaspora uvarum. So that comes in, that's very prevalent on grapes. And so that's present on all the grapes. So you would think that, you'd have the same sort of fermentation going on. So this may seem problematic to you. This may seem like, well, I added in RC212 and that's what I wanted to happen, right? That's what I wanted to do the fermentation and produce some fermentation aromas and things like that. I will say though, this is the sort of interesting, uncontrollable artistry, if you will, of wine, right? This is what gives us different wines, even if, 2 wineries got the same grapes, grape variety from one grower, let's say, they're going to make two very different wines. And just the, environment that those fermentations are happening in could really affect it. So while it may seem concerning, I would argue that maybe this is okay as long as you're not getting any off flavors and spoilage organisms. But there's an interesting interaction between these different yeast species. ICV-D254, which was not inoculated, seems to take over in a lot of these fermentations. Why is that? I'm gonna guess that the winery used that particular species a lot and was maybe floating around in the tanks, or was left over in tanks or things like that. Yeast can be very they can survive under unfavorable conditions for a while, and they just need they can easily kind of come back in and take over. So I just wanted to present that to show you that even though you're going to get a decent wine probably out of all four of these tanks, but they're not the same. They weren't fermented by the same microorganisms. So just keeping that in mind. So let's move into the controls. How can we control at least the growth of some of these microorganisms when we don't want them to grow? Okay, these 4 cases, these 4 tanks were fermented, so that we wanted growth of the microorganisms, right? We wanted them to uptake sugar and be active and be metabolizing. What are we going to do when we don't want those microorganisms to be active anymore? So, or before the fermentation. So one of the ways that I suggested was physical controls. So this is, there's some of these that we all take. Temperature, controlling temperature. So we do you can do a cold soak on red grapes, processed grapes. You could just keep grapes in a refrigerator, a refrigerated room or refrigerated truck before processing. You might keep your tanks cool before when you're doing a cold settling of your juices, or you might keep your tanks cool after the fermentation to not allow microorganisms to you really want to slow down their metabolism. If you slow down their metabolism, they can't grow and they can't make the organic compounds that give off flavors and things like that. So you really want to slow them down and prevent them from growing and metabolizing. There's also on the flip side of this, and there are wines that utilize pasteurization. I worked at a winery in France where they had issues in a particular vineyard, and they knew that there was a bacteria in that particular vineyard that they didn't want to rip out. It was a very old vineyard, and that was part of the story of the wine. But they would pasteurize the juice before they would do the fermentation because they really did want to make sure that that bacteria was taken care of and numbers were controlled. And there was no growth of it and things like that. So they did do a pasteurization on their juice. If you're making some other types of products like apple cider, sometimes there's even hard cider gets pasteurized. So, or no low alcohol wine is another kind of hot topic right now. And if you're exploring that, pasteurization may come up as a way for you to protect your wines before you package them. So you reduce, I will say pasteurization, the purpose of it is to reduce pathogens and to reduce, and on the example of, let's say, the no low alcohol wines, if you don't have that alcohol in there, you could have the chance of certain microorganisms being more viable or potentially pathogens. So you're really trying to reduce spoilage organisms and pathogens. You're not necessarily, it's usually high temperatures for relatively short time. So you're not necessarily completely sterilizing with pasteurization. That's the aim, is you want to sterilize as much as possible without deteriorating the quality of the product. And again, refrigeration, we use cold temperatures a lot, right, to inhibit microbial metabolism. Just know that if you are refrigerating your juices or wines, do bring them up to a decent temperature, 60 plus, I would say, Fahrenheit, before doing any filtration or racking or things like that. Because cold temperatures, you are going to pick up much more dissolved oxygen. So keep them cold when you're not working with those wines or juices. But if you do want to move them or do some sort of operation, filtration, like I said, make sure you do bring them up to a temperature that will limit that oxygen getting into those wines. Settling and filtration. So these are two things that we use to get rid of microorganisms. So we're going to and the settling part of it and the filtration part of it, quite frankly, is not just for getting rid of yeast and bacteria. It's getting rid of all, kind of particles that we don't want, all undissolved particles that we don't want in our wine. So it's a physical separation. Please know the difference. So settling, sorry, let me go back. Settling includes fining, if you want to use fining agents. So you add something into the wine. It binds to or interacts with some sort of compound, and typically it floats to, or not floats, but it settles to the bottom, and then you can remove your wine off the top. And then in terms of filtration, you're going to run it through some sort of filter. Please know the difference between absolute and nominal. If you are removing microorganisms, you need to have absolute filtration. And that means that all the pore sizes are the same. And you, for example, filter pads, the square pad filtration is nominal, okay? Meaning that if you have a nominal sheet, and it says that it's 0.45 micron, that means that the average size of the pores is 0.45 micron, not every single pore. Okay, some are going to be probably a little bit smaller, some are going to be a little bit bigger. So yeast could pass through those larger pores. Okay, so please know the difference between absolute and nominal. And if you are trying to do sterile filtration, you need to have absolute filtration, okay, or absolute filters, membrane filters, cross flow, things like that. Pressure is another way that we can control microorganisms, and you didn't use that a lot in the winemaking, but we do use it in terms of sparkling wine. Sparkling wine is fairly well protected, and that's because we build up a lot, I would say a lot of pressure. 90 psi, I think, is the typical pressure rating for champagne and traditional method winemaking or sparkling winemaking. So we do use pressure a little bit, not usually in our still wines though. Okay, moving on, chemical controls. So obviously sanitation. We are going to clean. I think most of us probably know that we spend, half our time, if not more than half our time, cleaning in our wineries. Please know the difference between cleaning and sanitizing. Cleaning is just the removal of dirt. Sanitizing is actually killing and killing, attenuating these microorganisms. So there's two different products for that. You clean with one product and you would sanitize with another. So please know the difference and that you do want to be doing those things on a very regular basis, and don't leave out things like, there's lots of nooks and crannies and tanks and valves, and when it comes to cleaning and sanitizing barrels, that's a whole other ball game, and I'm not going to get into that today, but please know that is a whole different ball game of what products you can use. But don't forget things like the floor and the walls and the forklift or the pallet jack. There's all kinds of things that can harbor microorganisms that could get into your juice or wine. So just remember, to be diligent. Okay, Jamie said with the chart. Thanks, Jimmy. Okay, so I'm going to jump into preservatives, okay? Sanitation, know your sanitation. Know that cleaning is not sanitizing. Preservatives, we use preservatives a lot. Obviously, we use sulfur dioxide a lot. There are wineries around the US, let's say, and even in Europe that are trying to get away from adding sulfur dioxide. And that's fine. Personally, I don't recommend that in our region. I think there's lots of risks of oxidation and bacterial and yeast growth within some of our wines. So I personally do not recommend doing sulfur dioxide non-added wines. They're not free wines. We could use sorbic acid as a yeast inhibitor as well. There is a legal limit to sorbate, so just know that. We can test it for you here at the Institute if you're worried about how much you might have in there. One that I just put on here because you might read about it is Velcorin. It's most of the time it's a big winery that's going to use that. I just saw on Scott Labs today, they were saying a proper Velcorin doser into wine is about $92,000 to get started. So this is obviously on the massive scale, probably that wineries are going to have a Velcorin machine. It's also extremely dangerous. Velcorin is like highly, highly toxic to humans, and it gets used up really quickly. Its half-life is really quick in wines, and that's why it kills everything and then it's gone really quickly. And so there's there's benefits to that, but it is super, super dangerous in the short term. Okay, the last one, or pH and ethanol. So ethanol is ethanol is a liquid, let's say, that we use for sanitation in the labs. You know, micro labs all over the university. What are they using to spray their bench tops down when they're cleaning? It's ethanol. They might use bleach as well, but a lot of them are using ethanol. So ethanol itself is just a good microorganism killer. Okay, there's some that are tolerant to it though, and those are the ones we have some problems with. pH. I wanted to make sure that I just, highlighted pH a little bit because I really feel like if you can understand pH and understand where we're at in the pH scale with wines and juices, and understand what a pKa means, and a PI maybe for proteins and things like that, you are going to understand so much more about the interactions of things happening in wine and the way that wine or juice, for that matter, can behave. And so I'm probably going to do a pH lunch and learn in the new year, because I just think it seems really basic. No pun intended, sorry. It seems like it's a really basic topic, but it's extremely important. If you really, really understand acid-base chemistry, you can really understand wine chemistry. Okay, so yes, because the pH is so low, most microorganisms do not survive at these low microorganisms. So that's pathogens especially, so we're good. So I used a couple of resources to look at the next section, and those are bioprotection strategies in winemaking. So using microorganisms themselves to protect your wine. And I know that sounds, why are we going to add in these microorganisms? They might do some bad things. If you do it in the right way, or if you're already doing it anyway with Saccharomyces cerevisiae, to be honest with you. So I just want to kind of walk you through it again to kind of wrap your head around what is bioprotection, and how could we potentially use it, or how are we already using it? So a couple of resources here, different papers, and then the OIV, the International Vine and Wine group in Europe, has a nice little document that they wrote this year. I think it came in March of this year. And so, yeah, and they've got some nice tables and things like that to go through of what different microorganisms you could use. Okay, so there's different strategies or different ways of thinking about this. So one is our passive strategies, and one is active strategy. So on the passive strategy side, this is all microorganisms do this. All microorganisms are uptaking different nutrients. And so if they uptake a nutrient, it's not available for another microorganism. So that's kind of the basic thought behind that. Or some of these microorganisms are they're secreting a chemical that uptakes a nutrient and takes it away from other microorganisms. And they're good at that. If they're good at this and they do it fast, then they can really prevent the growth and survival of other microorganisms. So also the kind of thinking along that line is oxygen competition. Now typically we talk about doing our fermentations in anaerobic conditions, so no oxygen present. So again, that's going to limit what microorganisms might be able to thrive in a solution if there's no oxygen present. And then the last one there is the space competition. And so I wrote down here biofilm formation. You might think, Erin, you're always telling us to clean and sanitize so we don't get biofilm buildup. Yes, you do not want biofilms in your wineries. However, on the surface of grapes, there are a couple of microorganisms that can form biofilms. They can kind of form these structures and all link together. And that protects the grapes themselves from the growth of other microorganisms if these biofilms are present. And you could potentially inoculate for that, but I don't actually know if that's if there are products available for that right now in the US. So I'm going to hesitate saying you can for sure do that. I'm not sure. I'm not sure if you can inoculate onto grape surfaces to protect them with a bioprotection method. I think so. There's more and more discussion about it. Let's say that. On the other side, we've got active strategies. So the production of antimicrobial compounds. And what are those? We've already talked about a couple: ethanol, SO2. Yeast naturally produce SO2 during, well, just in their metabolism. And so they do secrete that meaning they kick it out of their cells. And so when they kick it out of their cells, that can harm SO2 sensitive microorganisms. So they're actively protecting themselves. Ethanol, they're producing ethanol. They're not producing ethanol because they want to, that they're doing that for a whole different reason of redox potential and things like that. But the side use of that, the side benefit of that is that it keeps a lot of microorganisms at bay. There's a lot of antimicrobial peptides and proteins, which we call killer toxins, bacteriocins, lytic enzymes that certain microorganisms will produce and secrete, meaning they push them out of their cells so that they can affect other microorganisms. And so there's a whole, if you want to get into some of the resources that I mentioned that I was looking at, some of the papers, they go into details of some of these. I've heard that yeast can be killer yeast, and this is what they're talking about, these killer toxins. They produce proteins that will go and attack other microorganisms. Lytic enzymes, that means that they force cell walls to be leaky. And so when, just like you and me, if our outsides are, our barriers are leaky, that's not a very good thing. You want to keep everything intact. And so that can cause cell death and whatnot. Two interesting ones that I put down here as well: volatile organic compounds. So we think that, there's a lot of fermentation aromas that we get from during fermentation that the yeast are producing. They're either cleaving sugars off, or some of their metabolites are these volatile organic compounds, and they can be inhibitory to a lot of microorganisms. So some of the longer chain alcohols and things like that. So things that we don't usually think of in terms of bioprotection are there's evidence now that those things are actually helping us out in our winemaking. And then organic acid production, again, if you lower the pH or you're adding more acid, you can cause certain microorganisms not to be able to thrive. They have a real hard time in acidic conditions. So, and then cell-to-cell contact. There's some, especially Saccharomyces cerevisiae, that can aggregate. So as they flocculate, meaning at the end of fermentation, when they settle down to the bottom of the tank, they can join up together. They get sticky cell walls, drag other microorganisms along with them if they come in contact. So that's a good thing, right? They can kind of pick up other microorganisms and drag them down to the bottom of your tank. And then you can rack off or filter or do whatever you need to do. So active strategies, it depends what you're using, but Saccharomyces cerevisiae is pretty good at all of these. So, more benefit, it's high alcohol tolerance, it produces some nice aromas and things like that. And it also has some nice bioprotection. So we got lucky, I suppose, that we chose Saccharomyces cerevisiae to do our wine fermentations, and it just has all these other potential benefits. So this is just a diagram from one of those resources, and just talking about the passive strategies. And both yeasts or lactic acid bacteria tend to have or have these strategies, and it just depends on the different strains and the different species, which ones can participate in what. Okay. So I wanted to just put this in a little bit more perspective of what's actually available out there for us in the US, especially to use. Saccharomyces cerevisiae obviously is the yeast of choice for alcoholic fermentation. And there's some of its benefits. It does have fast nutrient uptake. You need to be careful with that because you do need to take into consideration the nitrogen requirements of your yeast. You don't want to start low and just hope your yeast fast uptake that, and then you don't have enough nutrient to finish the fermentation. So that's a bit of a balancing game there. They can coaggregate, they produce ethanol, they also produce some SO2, killer toxins, volatile organic compounds. So Saccharomyces cerevisiae is doing a pretty good job. There's a couple other species that are commercially available to add into your juice. You would probably add them. Most of them don't have high ethanol tolerance, so you use them at the beginning of your fermentation to get a couple of percent alcohol, and they have some other benefits as well. But in terms of bioprotection, here's some benefits of Torruspora delbrueckii. So you've got some fast nutrient uptake. It does produce a little bit of ethanol, and it's got some antimicrobial peptides. So it can, again, secrete those out and cause things not to thrive. Metschnickowia pulcherrima is really interesting. It's got a lot of benefits to it. So it can immobilize iron, which other microorganisms might need. Biofilm formation on grapes, on the outsides of grapes. Ethanol production, lytic enzymes, meaning those enzymes, it secretes enzymes, and then those enzymes make other cell walls of yeasts very leaky. And if things are leaking out, cells don't last very long. And VOCs, volatile organic compounds. And then Lacinaceae thermotolerans. It's got fast nutrient uptake, ethanol production, killer toxins, and volatile organic compounds. Enococcus eni is a lactic acid bacteria, obviously, that we use for malolactic fermentation, and it has quite a few different benefits as well. And just removing some of that malic acid makes and converting it into lactic acid, which is a less suitable metabolite, let's say, or food source, nutrient source for lactic acid bacteria, therefore making the wine more microbially stable. But it can also make some toxic compounds and does some biofilm formation and things like that. So some of the lactic acid bacteria are important as well. So again, these are some of the actual commercially available options from Scott Labs. So these are their names. There's Biodiva, Flavia Gea, Initia, and Lactea. However, so there's the Torruspora, the Metschnickowia, there's a different Metschnickowia, another Metschnickowia pulcherrima, and then a Lacinaceae. So know that they don't necessarily add all of that bioprotection that I did on the previous slide. They don't necessarily add all of that into their winemaking application or benefits, but know that they can provide some of those benefits. Some of them are here, like protects red grapes. This guy protects red grapes against microbial spoilage. This one protects white and rosé juice from oxidative damage and microbial spoilage. So especially if you're worried about maybe not getting to inoculation quickly, or you're worried about the state that the grapes came in on, or they came in, like if they were looking a little suspect, and using some of these might be not a bad idea. To keep other microorganisms at bay. Okay, so to wrap up, we do want to control the microorganism populations. I'm not suggesting that we just kind of let it all run wild. You can. Wild fermentations can be interesting. Um, Not always to consumers, though, is what I would say. I mean, it might be very interesting to you, but consumers might not quite appreciate wild fermentations. So we do want to control the microorganism populations. So there's a couple questions to ask. What is actually present? And we might not have, um, you might not have some of these spoilage microorganisms that you need to be careful of. You might be, they just might not be in your winery or in your wines or your grapes. Um, what is present? How can you control it? So knowing what strategies work best for, if you had Brettanomyces, what's a good strategy to use for Brettanomyces? Because it's SO2 tolerant, it's pretty ethanol tolerant. So which way should we go? What are you already doing to prevent those microorganisms? And is it working? It might be working. You might be on this going, Erin, I'm good. I've got a good protocol. I've got no Brett in my winery. I am pretty happy with what I'm doing, and I'm going to stay the course. And I would say, awesome. Like, I'm super proud of you for, coming up with good protocols and good cleaning techniques, I would assume. And yeah, being confident in that. But is there any room for improvement? Did you have one fermentation kind of get away from you? Or did you have one wine kind of go sideways one year? And what do you think happened there and whatnot? I will say that we have some new equipment in the lab where we can, if you're scared, you've got some sort of microbial spoilage, we can narrow it down a lot better now. We're not just looking under a microscope. We can confirm using genetic analysis that that you have a specific microorganism, whether it's a lactic acid bacteria or whether it's a specific yeast. So if you're worried about that, let us know. We'll work with you to do some testing. And the one thing this was from one of those resources, one of the reasons they were saying or suggesting to use these bioprotective methods is sustainability. And sustainability is kind of a buzzword at the moment, but we do need to start thinking about different ways to do things. Customers more and more are becoming educated, and they might not want high SO2 wines and you might need to, in terms of sustainability of your waste streams for your what's it called, your grape skins and things like that. So there's ways to think about where using some of these bioprotection methods at different stages in the winemaking process to improve on certain aspects. So down here, these are the impacts that you might see. So SO2 reduction are these little stick diagrams. And then there's microbial diversity, which is not always a bad thing, right? Remember when I was talking about those fermentations at the very beginning? Energy savings, there can be energy savings if you don't want to have to cool things down all the time, if you can control microbial populations in a different way. And then if you look at valorizing some of your waste streams and those grape skins and that sort of thing, there's a potential for value added, but also just being more environmentally friendly and that sort of thing. Okay, so all things to just kind of think about, in the winter as we get into a bit of a slower period, think about your processes, think about, is there anything I want to change? Where do I want to do some research? Just because you see one product or one commercial for a product, do your research, ask if this is right for you and your business plan, and reach out to us. We're happy to come and, chat through it with you and give you other considerations, and make sure that you're making the right decisions. Okay, so want to get into a discussion here. We're not going to record the discussion, but we will have this video up on YouTube shortly. But I don't like to record the discussion. That way you guys can all speak freely and we can chat about what you're doing in your winery, what you might want to try. But please reach out to us. We're happy to talk about this or any other winemaking topic. Check out our website. We're actively trying to improve our resources that are on there. Find us on YouTube, Facebook, Instagram. December 2nd, we're definitely going to talk about bottle closures. I haven't really talked about packaging at all yet, so we're going to talk about bottle closures. And then in 2025, send me your ideas. Happy and some great topics have come from winemakers and wine workers to, what do you want me to talk about? Maybe we'll do some panels or things like that and get other people up here. So you don't have to listen to me the whole time. And yeah, those are just some dates. First Monday of the month, going back, but there was an event happening last week. I didn't want to make people miss that event if they wanted to participate. So yeah, that is the first part of the month. If you're in Iowa, please consider coming to the Iowa Specialty Producers Conference in February. I believe it's February 11th and 12th. And if you're not in Iowa, check out your local conferences or come to Iowa. We're happy to have you. I'm sure we're going to have a really good, Christy always puts together a really good conference schedule. So yeah, I'm gonna leave this off here.